Everyone. Welcome to the Beijing Brain Conference, and welcome to our symposium. Our symposium is imaging-guided precision diagnosis and treatment of depression. So we know neuroimaging, it can be a prominent avenue towards the next generation of diagnosis or precise treatment for major depressive disorders. And we even can use brain imaging to explore brain imaging-based objective personalized diagnosis biomarker. And we want to use neuroimaging to localize precision modulation targets and develop imaging-guided personalized neuromodulation therapy. And the all symposium invited researchers from Harvard Medical School, Stanford School of Medicine, University of Toronto, Nanjing Medical University, and the Chinese Academy of Sciences to introduce the new frontiers of this field. And today, our first talk will be delivered by Dr. Michael Fox from Harvard Medical School. So Michael Fox is a founding director of the Center of Brain Circuits Therapeutics at the Brigham and Women's Hospital and associate professor of neurology at Harvard Medical School. He's also the inaugural Raymond Adams Distinguished Chair of Neurology and the King Family Research Director of Psychiatric Brain Stimulation. Dr. Fox completed a degree in electrical engineering at Ohio State University, an MD and PhD at Washington University in St. Louis, and a neurology residency and movement disorders fellowship at Massachusetts General Brigham Clinically. He specialized in the use of invasive and non-invasive brain stimulation for the treatment of neurological and psychiatric disease. Dr. Fox's research focused on developing new and improved treatments for brain disease by understanding brain circuits and the effects of neuromodulation. His papers have been cited over 39,000 times. Wow. And he was won awards across the fields of neurology, psychiatry, and brain stimulation. Honors include the inaugural Champion Prize for Clinician Scientists from the NIH, a single award across all medical specialties for advances in translational research. Today, Dr. Michael Fox will bring the talk using the human brain connectome to map and treat depression symptoms. Without further ado, let's please welcome Dr. Michael Fox to give his wonderful talk. Mike, please.
Thank you so much, Chao Gan, for the wonderful introduction and for the invitation. Thank you to you and the rest of the organizers. It's an honor to be here. Let me share my screen here.
Yeah.
All right. So it's can you see everything okay?
Yes, it's good.
All right. So it's my pleasure to kick off this wonderful, wonderful symposium full of brilliant speakers focused on optimizing brain stimulation for depression. So something that we think a lot about in my center is how we localize neuropsychiatric symptoms. And I think depression is a prime example. And our goal is to figure out the circuit or region in the brain that's responsible for a specific symptom. And for us, it's not just academic. We're very focused on therapeutic targets. So we don't want to just identify the spot in the brain that's responsible for donut craving or Duff beer, sleep, or depression. We want a brain stimulation target. We want a spot in the brain that we can implant electrodes to actually modulate the symptoms that we're trying to treat. Or a spot where we can hold our transcranial magnetic stimulation coil, again, to modulate the symptoms that we're trying to treat. And there are a lot of different valuable approaches for identifying these therapeutic targets. And I'd say the most obvious approach that's dominated my field for a long time is neuroimaging of symptomatic patients. It makes perfect sense. If you want to identify the spot in the brain that's responsible for depression, take a bunch of patients with depression, scan them with advanced scanning technology, and identify what brain region correlates with depression symptoms. Now, that has been a very, very powerful approach to understand depression and understand the brain. However, it doesn't necessarily translate into therapeutic targets. And the reason is that this traditional approach to functional imaging, no matter how many patients you scan or how much you increase the strength of your MRI scanner, you're still focused on symptom correlates, not causes of the symptom. And this is what contributes to what we call the causality gap. And we think it's very important if our goal is a target for brain stimulation. So in our center, we've kind of shifted our focus away from neuroimaging correlates of the symptoms that we want to treat to focus on causal sources of information that might be a more direct way to identify our therapeutic targets and the spots in the brain that we need to stimulate to provide symptom relief. And the most obvious causal source, or the traditional causal source for mapping out human brain function, is brain lesions. So when you have focal brain damage and a new onset neurological or psychiatric symptom like depression, there's a causal link between the spot in the brain that was damaged and that new onset depression symptom. And certainly we're not the first ones to take this approach. There's a very famous article by Robinson et al. In 1984 that found that lesions that hit the left dorsolateral prefrontal cortex are more likely to cause depression than lesions elsewhere in the brain. And by lesion, I mean a stroke, an ischemic lesion. And that was very influential. And in fact, that Robinson paper is what led to the first TMS trials trying to excite the left dorsolateral prefrontal cortex to help depression. The problem is that this hasn't always replicated. So some papers did show this association. Many other papers did not show this association. And this is just one example of a meta-analysis in 2015 of over 5,000 lesions. And they didn't find any support that lesions to the left hemisphere or the left dorsolateral prefrontal cortex were associated with an increased risk of depression. And this is a common problem in lesion-based localization. It's not just depression, it's many other symptoms that we've tried to localize based on focal brain damage. So this is how lesion-based localization is supposed to work. You take 3 patients, patient 1, patient 2, patient 3, all of whom have focal brain damage and a new onset symptom, let's say depression. You map out the location of the brain damage in each of these patients onto a common brain atlas, and you see where in the brain do these lesion locations intersect. And when Robinson did this in 1984, he found that they did intersect in the left dorsolateral prefrontal cortex, but then other people didn't find that. And that's what we've seen over and over again with lesion-based studies, is that you take 3 patients that all have focal brain damage, all 3 of these patients have the same new symptom, and all of them have a causal link between their focal brain damage and that new symptom, but you're left scratching your head because the lesions aren't in the same spot, then they don't overlap any single brain region. Well, that's when we can turn to a new resource called the Human Brain Connectome. This is a wiring diagram of the human brain that's been built as a collaborative effort across the neuroimaging community over the last few decades. And we have an anatomical wiring diagram of the human brain, so where the major white matter connections are. We also have a functional wiring diagram of the brain. That's what you see there on the right. That's brain regions that are correlated based on ongoing patterns of spontaneous activity. So let's turn back to our problem of lesion-based localization. We know that for each one of these patients, there's a causal link between the focal brain damage and that new onset symptom. But now, rather than ask, do these lesions intersect one brain region, for the first time, we can use the human connectome to ask whether or not these lesion locations intersect a common brain circuit. And this has been a very, very powerful approach for mapping out neurological or psychiatric symptoms. And so this is just a running list of symptoms that my particular lab and center is mapped out based on lesion damage. And in each one of these cases, the lesions did not map to a single brain region, but they did map to a connected brain circuit. And we're not the only ones finding this. There's actually 6 or 7 other labs that have been mapped out a variety of other neurological and psychiatric symptoms. But today we're just going to focus in on depression. And so Jaya Padmanabhan in the lab worked very, very hard to look at this question of where do lesions need to occur to cause depression. And it ended up taking 5 different data sets across 4 different continents to go ahead and get an answer. And in the end, she ended up identifying about 60 lesions that cause new onset depression and about 300 lesions from those same data sets that cause no depression symptoms at all. And the first thing she did was the traditional approach, just like Robinson, to say, okay, is there any spot in the brain that when lesioned causes depression? And she didn't find anything. Even when she did an a priori region of interest in the left dorsolateral prefrontal cortex, there was no statistical association between lesions to any specific spot in the brain and depression. However, she then used the human connectome to look at what each one of these lesion locations was connected to. And then she repeated the analysis, but this time she said, are there any connections with the lesion location significantly associated with depression? And now she got a very strong hit, again, to the left dorsolateral prefrontal cortex. So lesions didn't have to hit that region, but they did have to be connected to that region. Now, what that means is you can go to this one spot in the brain, the left dorsolateral prefrontal cortex, look at what it's connected to, and that defines a distributed brain circuit that when lesioned will lead to depression. And so you can see that, yeah, if you lesion the left dorsolateral prefrontal cortex, that will increase your risk of depression, but also the right dorsolateral prefrontal cortex, also parietal regions, also temporal regions. There's even spots in the cerebellum that when lesion should increase your risk of depression, and a whole lot of other regions that when lesion might actually protect against depression. And in fact, this seemed to line up pretty well with TMS treatments that have been used specifically for post-stroke depression. And that's what those 3 dots are in the left dorsolateral prefrontal cortex. Those were 3 trials where they did excitatory TMS to treat post-stroke depression, and all 3 of those trials were positive. Now, the reason that's important is it suggests that we can use lesions that cause a symptom to identify a circuit, and that might be an ideal circuit target for TMS to provide symptom relief. Now, we can also turn to another causal source of information, which is brain stimulation itself. And just like lesions give us a causal link between the damaged brain tissue and a symptom, brain stimulation gives us a causal link between the stimulation site and a symptom change. And so let's just focus in on TMS, because we can actually look at the heterogeneity in where our patients were stimulated. So not every patient gets stimulated at exactly the same spot, because we target TMS using scalp measurements. And we can just look and say, okay, who got better and who didn't get better? Now, in this case, we had an a priori hypothesis. We thought that the limbic region, specifically the subgenual cingulate, was playing a role in antidepressant response. And we said, okay, here are the different locations that these different patients are stimulated. Are the patients that are stimulated at a site that's connected to the limbic system, are those the patients that are getting better? And that's exactly what we found. The more connected your stimulation site was to the subgenual cingulate, the better your antidepressant response. And what was very exciting to us is unbeknownst to us, at the same time we were doing our experiment, Robin Cash and Paul Fitzgerald were doing the exact same experiment in Australia, and they saw the exact same thing. The more connected your stimulation site was to the limbic system, the better your antidepressant response. So this has led to the idea that you might be able to use brain connectivity with the limbic system, specifically the subgenual cingulate, see what that spot in the brain is connected to, and that might be your ideal target to deliver TMS, to make depression better. And in fact, there's some evidence that that actually is an effective strategy. So you're going to hear from Nolan Williams in just a few minutes, who's going to tell you about his really exciting work targeting TMS based on individual connectivity to the subgenual cingulate and getting an 80 to 90% response rate. But he's not the only one. There's other people that have targeted this anti-subgenual node. In fact, Dan Bloomberger, that you're going to hear about after Nolan Williams, used this target in his landmark 3D trial. And so, that trial is all about 10 hertz versus theta burst. But one of the things that gets missed is everybody in that trial was stimulated at this anti-subgenual target. But the problem is they still saw a 50% response rate and a 30% remission rate. That's about what we saw with the old scalp-based targeting. And if this was the whole answer, then I would have expected to see the response rates going up a little bit. And even more concerning was this paper that came out in JAMA Psychiatry, where, again, they didn't target exactly our coordinate, but they still targeted roughly our coordinate. And they saw a trend of, active better than being better than sham for depression, but these were all patients that had depression and PTSD. And there was actually a trend towards it being worse than sham stimulation for PTSD symptoms. And these studies really made us stop and think and say, okay, if we're right, if this is all there is to targeting TMS for depression, we would have expected to see a little bit more signal in these big trials. And so Sean Siddiqui decided to go after this question and said, okay, well, maybe there's not one target for depression. Maybe there's different targets for different symptoms of depression. And so what Sean did is he used a data-driven approach and said, okay, what are the connections that we need to hit to improve sadness or anhedonia or concentration? And what are the connections we need to hit to improve other symptoms of depression, like decreased interest in sex, sleep abnormalities, indecisiveness? And what he found is there were very different connections you needed to hit to improve different symptoms of depression. But some of them all looked the same. And what we found is that there were actually two different circuits that accounted for two different clusters of depression symptoms. There was one circuit you needed to hit if you wanted to improve dysphoric symptoms of depression, and a totally different circuit you needed to hit if you needed to improve anxiosomatic symptoms of depression. And that's actually led to a randomized controlled trial that we're doing right now, targeting circuit A to see if that helps one set of depression symptoms, and targeting circuit B to see if that helps a different set of depression symptoms. But the idea is that there might actually be circuit targets that are different depending on what symptoms bother the patient. And in fact, that's exactly what we do in deep brain stimulation for Parkinson's disease. We use different targets depending on the symptoms that bother that particular patient. Another possibility is that it's not just the subgenual cingulate. So one of the things that we've done recently is what we call convergent causal mapping, where we say, hey, brain lesions give us one unique window into what brain circuit might be causing a symptom. And brain stimulation gives us another unique window into what brain circuit we need to manipulate to fix a particular symptom. But what if we put all causal sources of information together? And that's exactly what Sean Siddiqui did in another paper of his. He found every lesion, actually the same lesions that you saw from Jaya Padmanabhan's paper. So 5 cohorts, 465 lesions, everybody had a depression score. He found 4 cohorts of TMS patients with exact records of where they stimulated, 151 stimulation sites, and what happened with depression symptoms. And then he found 101 deep brain stimulation sites across 5 cohorts, and what happened to their depression symptoms. And he said, okay, I can use lesions, I can use TMS, I can use DBS. And in each one of these data types, I can map out what are the connections that you need to hit for a lesion to cause depression? What are the connections you need to hit for TMS to fix depression? And what are the connections you need to hit for DBS to either make depression better or make depression worse? And it was the same circuit. No matter whether you derived a depression circuit using lesions or TMS or DBS, these 3 causal sources of information all converged on the same brain circuit for depression. And so we can put those 3 different causal sources of information together and say, this is our brain circuit target for depression. And you can see the subgenual cingulate is in this circuit. That's that green region down there. But there's a whole lot of other regions in this circuit. It's a circuit target, not just one region. And you could show that if you target TMS or DBS using this circuit, using a leave one data set out analysis, we're able to predict antidepressant response based on how well your TMS or DBS electrode hit this circuit. And in fact, this circuit worked better than the subgenual cingulate for predicting antidepressant response. So I'm going to leave you with 3 conclusions. Conclusion number 1 is that lesions, DBS sites, and TMS sites allow for causal links between human neuroanatomy and symptoms. take-home point number 2 is that a map of human-brain connectivity is required to make sense of this data. All the lesions, DBS sites, and TMS sites are in different locations. And then take-home point number 3 is that by leveraging the human connectome, this wiring diagram of the human brain, we can identify the causal substrate of depression symptoms, and we hope better therapeutic targets for brain stimulation to provide symptom relief. And I want to thank my lab, and I thank them as we went along, but I also want to thank my worldwide network of collaborators. I did not collect the vast majority of data that you saw presented here. And I am very, very grateful for all of the centers that contributed their data and all the centers that worked with me to understand that data to do the work that I showed. I also want to thank our growing network at the Center for Brain Circuit Therapeutics. It's our unique collection of neurologists, psychiatrists, radiologists, and neurosurgeons all working together to try to use brain circuits and brain circuit imaging to make our treatments better. And at this point, I hope I left some time for questions. Thank you so much.
Thank you very much. Platform. Let me take the advantage that ask you some questions. So first one is, I saw your circuit map. I also noticed maybe the lateral parietal or maybe near the angular gyrus is also a very prominent area. And we also know angular gyrus may have connection to hippocampus or parahippocampus, connection with the memory. So actually our lab is kind of concerned on the medial temporal lobe, some related memory, and the angular gyrus seems to be better than dorsal prefrontal cortex to them. But do you aware any treatment on the angular gyrus or lateral parietal to treat depression?
Yeah, excellent question. So first, I do want to separate out the anatomy of the parietal cortex. So the angular gyrus is usually that default mode network node that's connected to the hippocampus. That's what Joel Voss has stimulated to try and modulate memory. And it sounds like that's what you're also going after. That's actually a different spot. So that big yellow area that you saw jump out, which we predict might be a very effective target for high frequency stimulation, right, is actually in the interparietal sulcus. Kind of part of the dorsal attention network. And so that's different than the angular gyrus, right? It's actually the opposite network.
The opposite.
But you're totally right. Our number one peak in our depression circuit when we put all 14 data sets together was not the dorsolateral prefrontal cortex. It was the interparietal sulcus. And I do have colleagues that are doing trials now targeting the interparietal sulcus with high frequency stimulation to see if that might be an effective treatment. But I only know of one lab that's doing it. And so I would love to see more labs try that. So if anybody's listening to this talk right now, go after the interparietal sulcus and let me know what happens, because I actually do think that's a very promising target that comes out of our data. But people are just now beginning to explore that as a depression treatment target. Also happy to send you the map or send you the coordinates if you want to try and target this circuit for antidepressant response.
Another related question is because allow me so many questions that we know most people is doing the left dorsolateral prefrontal cortex, but some people also use a low frequency on the right DIPFC, and some people like Cornelis, I think, you target on the DMPFC, medial prefrontal cortex. Do you have any comments on, at least on these three prefrontal targets?
Yeah, great question. So I'll start with the low frequency right. So the short answer is that when we derive a depression circuit, the left dorsolateral prefrontal cortex is a brighter node than the right dorsolateral prefrontal cortex. However, they're both in there and they both have the same sign. So our circuit maps do not provide clear justification for doing low frequency right. That being said, the clinical data does suggest that low frequency right can be effective. So how do you think about that? And the way that I think about it is that our idea that you have excitatory high frequency stimulation and inhibitory low frequency stimulation might be kind of true in primary motor cortex, but it might not be relevant for prefrontal cortex or for depression. It might be that all you need to do is whack the correct circuit, and you can whack it with high frequency, you can whack it with theta burst, you can whack it with low frequency, but if you deliver enough pulses, that can be effective at making depression better. And we actually have very little evidence that low frequency stimulation to the left doesn't help, or that high frequency stimulation to the right doesn't help, right? There's only 2 or 3 studies that have ever suggested that's true. So I think it might be that you just need to deliver TMS pulses to the depression circuit, and that this high frequency left, low frequency right might not actually be as meaningful as we think that it might be. Now, the dorsal medial prefrontal, that's a very different circuit. And that's the circuit that we are trying with high frequency stimulation to try and help anxiosomatic symptoms of depression. So our thought is that it might be that different circuit targets, the lateral prefrontal cortex versus the medial prefrontal cortex, you're definitely hitting different circuits, and that might be helping you treat different symptoms.
Okay, thank you very much. So I saw a question from the audience, also related. So will there be different effects if stimulating different sides of the same circuit?
Yeah, great question. The short answer is we don't know. So one of the big limitations of functional connectivity is that you get these polysynaptic circuits, right? That's a strength because you need something that is sensitive to polysynaptic connections to link up these lesions, TMS sites, and DBS sites that are all over the brain. However, when you get a circuit answer, right, you get multiple nodes. And so we have no idea if targeting the left dorsolateral prefrontal cortex or the interparietal sulcus is better at actually modulating the circuit. And so 2 things that would help us in that regard. One is just experimental evidence. Run the trial, target both nodes, and see if they both are capable of modulating the same circuit, because I don't know the answer. The other one is to use newer imaging methods to try and get at causality. So functional connectivity just gives you correlation. So all we know is that our lesion locations, our stimulation sites are functionally connected to this circuit, but we don't know anything about the causal direction of information flow. And it might be that if you do Granger causality or use a connectivity map build off of MEG or build off of ECOG or ECOG stimulation, it might be that different sites in the same correlated circuit have a larger causal effect on the circuit target than other sites.
Okay, so Daniel or Fei, would you like to take the advantage to ask? We only have 2 minutes. You ask some questions. Daniel and Fei.
Hi, thank you, Togan. Dr. Fox, I already read your paper publications millions of times. We are following you. But one question, the symptoms may be related with this circuitry you talk about millions of times. But how do you think about this circuitry for diagnosis?
Yeah, it's a great question. So we have a commentary that Sean Siddiqui and Joe Taylor, Andy Horn, and I just put out in Biological Psychiatry of how we use connectivity in psychiatry, and there we kind of we made the argument that we've been trying to use functional connectivity for diagnosis for 3 or 4 decades now, and we haven't made a huge amount of progress. And so it doesn't mean that we won't get there. But for us, we've decided that in the end, as clinicians, my patients don't really want a diagnosis, nor do they really care what their diagnosis is. And in psychiatry, diagnoses are very difficult, and most patients end up with 4 or 5 different diagnoses because it's a checklist. And so I think what people really want, right, is not necessarily a diagnosis, but they want something that will tell them what treatment is going to make them better. right? So right now you give people a diagnosis of depression. That means, oh, you should try this class of medications, and you should try this type of brain stimulation targeting this circuit. But for us, you can almost sidestep the whole question of diagnosis and say, hey, nobody really cares what their label is. What they care about is where they need to be stimulated to get them feeling better. And so I'm hoping that we might reach a point in psychiatry or neurology, for that matter, where we can say, okay, you have a disorder of brain circuit A. So we're going to stimulate brain circuit A. And you have a disorder of brain circuit B. We're going to stimulate brain circuit B. And what combination of current labels are you the depression, PTSD, OCD type? Okay, that's brain circuit, you know. And so I guess for us, I try to avoid the very difficult problem of diagnosis and skip straight to treatment.
Great idea. Yes, we don't care about the boundary. We just, if we can help them. Okay, great. Thank you.
Okay.
Yeah, great question, though.
Mac, thank you very much for your wonderful talk and the answers. And I think you always lead the frontiers, and we do think that the circuit we should follow, and we should target to help the patients in the future. And many thanks to your talk. And if the audience have any further questions, you can ask Mike, maybe in email, or maybe some of us will, at the end of the symposium, we can have some short discussion. But Mike, we thank you very much for your talk, and we will move to the next one.
Thank you so much for having me, and thank you for the brilliant questions.
Thank you. So, because Nolan has something need to deal with, and we'll attend our symposium later, so let's move Dr. Daniel Bromberg to the talk, move ahead. Subspecialist in generic psychiatry. He's a Trinity Chair in Therapeutic Brain Intervention and the Director of the Trinity Center for the Therapeutic Brain Intervention. Dr. Bromberg is a professor in the Department of Psychiatry at the University of Toronto. His primary area of research focuses on clinical intervention trials of novel treatments for depression and other psychiatric disorders across the lifespan, using brain stimulation therapies. In addition, his research aims to better understand the neurophysiology of mental illness using state-of-the-art methodologies, including TMS and EEG. He's a principal of co-investigators on multiple clinical trials using different brain stimulation modalities, including magnetic seizure therapy and repetitive TMS. He will bring a talk on pathways to personalize RTMS delivery for depression. So without further ado, let's welcome Dr. Daniel Bromberg. Hi, Daniel. See.
Great. Okay. So once again, thank you for having me, and wonderful to be here amongst these great speakers. Hopefully Nolan will be able to join, and it's great to hear Mike's talk. And I'm glad I didn't pull too much of his seminal work into this talk. So here are my disclosures and funding sources and connections with commercial interests. But when we're talking about personalized RTMS delivery, we're talking about individualized delivery of repetitive transcranial magnetic stimulation, and it might involve various different modifications to how we currently deliver treatment. It might involve modifying a different region or a different circuit, modifying the protocol or the dose of the treatment, the scheduling of treatments, combining RTMS with other techniques or other potential personalization methods. The problems that we have now are the inconsistencies across multiple studies and the way variables are defined and measured. The protocols are extremely heterogeneous across studies. Inclusion criteria are different, And the work surrounding predictors are mainly retrospective and involve smaller sample sizes. So we need more data-driven multimodal approaches to improve the likelihood that we're going to get towards personalization. And the benefit of personalized treatment is that we'll more efficiently and effectively treat those suffering from depression, and will reduce the high costs of treatment delivery. So here's an outline. We'll go through some of the clinical and demographic predictors. So what has been done in terms of just looking at the patient in front of you and the characteristics that they present with in terms of their depression and in terms of their demographics. And then we'll go through some of the biological predictors. So looking at structural and functional connectivity imaging, electrical field modeling, and we'll talk a little bit about brain oscillation triggered TMS. So looking at also what is happening within the brain and triggering the stimulation to the ongoing oscillations occurring within the brain. And then we'll talk a little bit about what might a personalized RTMS treatment look like. So in terms of clinical predictors, there's quite a bit of variability in response rates following TMS. And this may be attributed to many factors: someone's age, their sex, their employment status, brain morphology, disease-specific factors, including the duration of the episode, the severity of their depression, which can encompass suicidal ideation and suicide risk, the number of hospitalizations, the type of antidepressant that they've had, the amount of treatment that they've had, and their comorbid anxiety symptoms, and whether or not there's a presence of a personality disorder. The current method of delivering RTMS are implemented via trial and error, and there's a lack of replicable and quantifiable predictors. But there are some hints in terms of what clinical features lead to a better response to TMS. So this work out of Paul Fitzgerald's group, they looked at 11 of their clinical trials and over 1,100 patients. And compared to non-responders, responders were interestingly older, had lower depression severity, and had a shorter illness duration and a later age of onset of their depression. And response was more likely in single versus repeated episodes of depression. And it was more common for patients receiving an antidepressant at the time of treatment, and more common in patients with no comorbidity than those with anxiety comorbidity. And this was primarily treatment delivered at high frequency stimulation to the left dorsolateral prefrontal cortex. We looked at the data in the 3D study, and we wanted to really focus in on remission, which is really the outcome that matters most to patients. So whether or not they're going to get fully better in terms of their depression. And we took some of the variables that the Fitzgerald group came up with, including age. We looked at employment status as a measure of their functionality. We looked at the baseline depression severity using the HAM-D. And we used the degree of treatment resistance using the antidepressant treatment history form as a marker of their severity of their illness and the duration of their treatment resistance. And we put these predictors into a logistic regression model. And we came up with a model that actually did reasonably well, but not at the level where you could just look at a patient, look how old they were, look at how severe their depression was, and say, you have greater than 50% chance of responding to this treatment. So in terms of prediction models, we had a C statistic that was 0.687, which was moderate. You want to get to a prediction model that has greater than 0.7 in terms of having any utility in terms of predicting outcomes. But with these 5 variables, we were able to come up with a relatively decent ability to predict outcomes and predict the most important outcome, which is remission from the depression. So if we can find some other biological variables to combine with these clinical and demographic features, we may get to a point in terms of being able to better predict who's going to respond to treatment. So what might those biological predictors might be? So you heard from Mike about the connectivity target in terms of the left dorsolateral prefrontal cortex and the subgenual cingulate. And this is work that has been done out of Australia, Robin Cash, Paul Fitzgerald, and Zalesky, looked at outcomes when stimulation was delivered at the sites of the DLPFC most anticorrelated with the subgenual cingulate. But there's a high degree of inter-individual variation. So they wanted to look at scans from 1,000 healthy individuals and to see whether or not they could come up with a method of targeting, using this method to target the dorsolateral prefrontal cortex and the anticorrelated site in the SGC. And what they found was that the topography of the SGC across the DLPFC was consistent within individuals, but highly variable between individuals. So personalized target had an accuracy of about 2 millimeters between scans and remained stable after 1 year. So these are things that you want to see in a potential predictor of clinical outcome. And the inter-individual variation suggesting that personalized targets did not trivially converge to a group average site. And this may explain to some reason why we didn't see those outcomes that we wanted to see in the 3D study. Even though we used Mike's target, we used a group average rather than an individualized average. And they looked at this outcome and they looked at functional connectivity between the SGC and the DLPFC. And again, the target most anticorrelated with SGC were clustered. And the closer proximity that the coil was placed to that target led to better clinical response. And Sean and Mike also replicated this as well in a longer treatment duration study out to 6 weeks instead of 3 weeks. So again, the closer you are to the target, the better the outcome. And interestingly, this work, so in terms of targeting the SGC, can also potentially be done using a structural scan. So this is Sean Nestor, who's a junior clinician scientist that I work with, wanted to see whether structural network integrity and plasticity were associated with response to TMS using the 3D data set. So they looked at structural MRIs from 70 healthy controls and 268 of the patients with depression in the 3D study. And he was able to find that the cortical thickness and volume within in these structural covariance networks distinguish healthy controls from depressed, but there was no association between the structural covariance network expression and their clinical improvement. However, an interesting finding from this study was that they found the anticorrelated structural covariance network between the left dorsolateral prefrontal cortex and the subgenual cingulate was found across cortical measures. And post-treatment change in the cortical thickness of this covariance network was associated with clinical improvement. So potentially this finding needs to be replicated, but potentially you could take a structural scan rather than a functional scan and potentially use the same mechanisms of personalizing and targeting. You could imagine that a structural scan would be a lot cheaper and a lot easier to get than a functional scan. So we've gone through some functional and structural connectivity data. Now, it's important to be able to identify where in space you're stimulating the target. But there's a very important component to being able to deliver TMS effectively to that target within the DLPFC. And that is the actual morphology of the gyri and the sulci induced in the electrical fields underneath the coil. And there's a great degree of variability in the structural anatomy of each individual. And the way you place the coil impacts the electrical field and whether or not you're actually going to be able to hit the target. So neuronavigation can be quite effective in the motor cortex, where you have a very straight area and a very easy target. But within the dorsolateral prefrontal cortex, we don't know which way the target is and where it's hiding within the gyri. So TMS coil positionality and targeting can lead to marked variations in the E field distribution and intensity, and it can really impact MEPs. So you can imagine the DLPFC, you could be placing your coil in a standardized way, but this may not be targeting the area within the circuit that you want to hit. So Engel Peterchev's group has come up with a method of being able to improve the accuracy in coil positioning with respect to using a fast computational auxiliary dipole method. So they've optimized coil placement that maximizes the E field along the predetermined direction of the overall E field magnitude in the target region of interest. So this method, this ADM method, computes the induced E field generated by a virtual constant current source residing within the region of interest. And what they found is they could get to the regions of interest below 2 centimeters with an average magnitude of the E field can be estimated to an error below 2%. So the system allows for a rapid extraction of the optimal coil position to be able to optimally target the area within the dorsolateral prefrontal cortex or the area underneath the coil. And it allows one to quantify the uncertainty in the induced E field due to limited precision of TMS coil placements. And it allows minimization and statistical analysis of the E field dose variability. So we're getting to be able to actually target the areas that we want to within the dorsolateral prefrontal cortex, not just by structural imaging, but being able to model and position the coil to optimally target that area underneath the coil. So this is Daphne Vaneskos, who's another clinician scientist that I work with, who's going to do this to be able to look at E-field modeling and individualized connectivity-guided TMS to see whether or not these, if one is able to highly individualize both the E-field and the connectivity delivered to TMS, ITBS, is superior to just the standard method of using Beam F3. And she'll also look at this in terms of clinical outcomes and cortical excitation with TMS EEG. And this will be an adequately powered clinical trial, randomized clinical trial, without 6 weeks of duration to really see whether or not this optimized method of targeting this functional target, as well as an E-field modeled delivery of the functional target, is superior to the standard method. And these are the kind of trials that we need to be able to see whether or not this personalized approach will get us towards improved outcomes. So we've been talking a lot about space. Now, what about time? The brain is not a static target, it's a moving target. Our brains are constantly moving while we're thinking, while we're speaking, and while we're experiencing emotions. So this is where Eeg Triggered Tms comes in. So closed loop stimulation uses the patient's own physiological activity to trigger stimulation when a pathological state is detected. And it synchronizes the timing of TMS stimulation with the phase of the ongoing EEG. And TMS synchronized to surface EEG at the negative peak of the sensory motor alpha rhythm can lead to a differential corticospinal excitability and plasticity effects. So this may underscore why we see all these variable outcomes, because in certain individuals, we may be stimulating at a different phase of their ongoing brain oscillatory activity. So this is Christoph Renner, who's pioneered a lot of this work in Ulf Siemens' lab in Tübingen, who's now come over to Toronto. Really wanted to see whether or not you can not only look at the alpha rhythm, but also the theta rhythm. So he developed a method to apply TMS phase-locked ongoing theta oscillations in the left dorsomedial prefrontal cortex. And they extracted EEG signals to simulate real-time phase detection and quantify the accuracy versus post-hoc phase estimates. And what they found was that using optimized parameters, the prefrontal theta phase synchronized TMS in the DMPSC they were able to achieve it with a high degree of accuracy. And Christoph also has a spouse, and Bridget, his spouse, has pioneered this work in depression as well. So using this real-time brain oscillation synchronized TMS in a single session in the left DLPSC in 17 patients, they were able to hit the negative peak of the instantaneous alpha oscillation. And they were able to synchronize the alpha synchronized TMS, but not the other conditions, led to a reduced resting state alpha in the left DLPSC and increased TMS-induced beta oscillations over the frontal-central channels. So the targeted alpha-synchronized TMS led to an altered response neurophysiologically. And this is some work so coming out looking at daily prefrontal closed loop TMS using a EEG quasi-alpha phase entrainment in depressed adults. So they were able to, the objective of this work was to determine if TMS brain dynamics dependent on TMS relative to individuals' prefrontal alpha rhythm in depressed patients. So they were able to lock on to the phase of the synchronized alpha rhythm and looked at specifically the strongest fMRI activation in the subgenual anterior cingulate. And what they found was that the TMS applied over the DLPFC and synchronized patients developed a strong phase entrainment over a period of weeks over the stimulation site and distal areas. And at the end of the treatment course, only the synchronized groups entrainment shifted closer to the phase that optimally engages the anterior cingulate. And the entrainment effects built over time, suggesting that they engaged neuroplastic changes that may have clinical consequences. And it may be that the way that they engaged the DLPFC and the subgenual cingulate in this study may have had a counterintuitive effect and may have led to different outcomes in terms of depression outcomes. All this to say that this work with respect to targeting TMS delivery to the ongoing oscillatory state within the brain is a very powerful and potentially important approach that allows us to be able to get to a more personalized delivery, but we need a lot more work to be able to figure out whether or not at what phase of the stimulation we need to be delivering our treatment. So in terms of the summary, the number of retrospective studies looking at clinical predictors highlight the need for selecting the right patient for the treatment. And retrospective studies suggest that closer stimulus to the closer the stimulus in the DLPFC location that is anti-correlated with the SGC results in a higher likelihood of response to TMS. But we need prospective trials, and a number of these trials are ongoing. And it'll be very fascinating to see within the next 3 to 5 years what the outcome of these personalized connectivity-guided trials yields. And ensuring that the stimulus hits the intended target within the DLPFC can be achieved through E-field modeling. So I believe that the being able to ensure that one is not only hitting the structural target, but being able to hit it with the correct E-field is an essential part of this personalization approach. And the variability in outcomes may also be related to individual brain oscillation dynamics in relation to the type of TMS delivery. And real-time brain oscillation synchronized TMS may also lead to improved outcomes in terms of getting to personalization. So I want to talk about the brain's space-time continuum. So the brain is, people have said, is more complex than the universe. But the universe exists in not only space, but also time. So we need to be able to pick the right patient. We need to be able to hit circuit correctly using E-field modeling. But we may also need to be hitting the correct circuit using the correct approach, but at the right time. So being able to target our oscillation to natural oscillations that are occurring within the brain may be an important pathway towards personalization. So we have the great privilege to work with a great team of students, trainees, clinicians, scientists, clinicians, and thank you for paying attention, and to all the collaborators that I work with as well. And hopefully we have some time for some questions.
Thank you, Daniel. Very wonderful talk. ground level, then how to perform a real-time EEG triggered TMS?
Sorry, can you repeat that? I didn't see the question.
Is in the chat. So
okay,
it's that the TMS pulse will affect EEG signal, and it may take time to return to the ground level, or maybe base level. So then
correct.
how to perform the real-time EEG triggered TMS?
Right. So it's a very important question. I would have to say that the technology and the physics behind being able to target TMS in real time requires a specified and special system to be able have to deal with that return of the ground level.
Okay. Actually, myself, I do have a question that
yes.
So you're talking about space, about time, and I also think about task. So actually, in our lab, Xiaocheng's work is doing on the illumination. So we identify some illumination circuit. So we are wondering if we target this illumination circuit during the patients, they are actually illuminating. So combining space, time, and task, what's your opinion?
So that's a fantastic question, Shogun. And I think that is our window into being able to hit the circuit in the right time, being able to activate the circuit. So part of the work in terms of real-time TMS EEG is being able to activate the circuit and being able to see the circuit. So with rumination, I think you can provoke ruminations. There are certain symptoms that you can provoke. You can provoke anxiety relatively well. You can provoke obsessions. There's a very interesting study out of University of California, San Francisco, that did this using deep brain stimulation in a depressed patient. So I think that when we talk about time, figuring out the brain state and activating the brain state is an important aspect of understanding time.
Okay, thank you. Then another question comes from the audience is that will you consider other combinations of techniques such as fMRI TMS to stimulate certain circuits?
Another fantastic question. So, potentially combining fMRI with TMS and EEG will actually be able to allow us to see both space and time in the same place. There are some people working on being able to use a closed loop real time within an fMRI scanner, within an MRI scanner. So I think that is probably the highest level of precision with respect to being able to, so we would be able to stimulate specifically while the brain is activated, while viewing the EEG and the circuitry in real time.
Thanks. Mike, Faye, and Nolan, would you like to take the advantage to ask some questions?
Hey, thanks, Stan, for switching me. It was a last minute switch, so I really appreciate you doing that.
No problem.
Yeah. Very cool. Yeah, I caught the tail end of that. It's interesting. I've been talking about the S&T or Saint protocol as a reorganization in space and time as well. So we're all thinking in the same in that same vein. So very, very, very cool. What I caught was great. So
thanks, Don.
Okay. So thanks, Daniel. So thank you very much for your wonderful talk. So if the audience have other questions, they can send you email or we can save some time. Brought by Dr. Nolan Williams. Dr. Nolan Williams is an assistant professor within the Department of Psychiatry and Behavioral Sciences, and director of the Stanford Brain Stimulation Lab. Dr. Williams has a broad background in clinical neuroscience and is a triple board certified in general neurology, general psychiatry, as well as behavioral neurology and neuropsychiatry. Themes of his work include examining the use of space learning theory in the application of neurostimulation techniques, development and mechanistic understandings of rapid-acting antidepressants, and identifying objective biomarkers that predict neuromodulation responses in treatment-resistant neuropsychiatric conditions. He has published papers in high-impact peer-reviewed journals, including Brain, American Journal of Psychiatry, and PNS. Results from his studies have gained widespread attention in journals such as Science and the New England Journal of Medicine. And he has lots of awards, like 2 NASA Young Investigator Awards in 2016 and 2018, along with the 2019 Gerard Clement Award. So Dr. Williams received the National Institute of Mental Health Behavioral Research Award for Innovation Neuroscientist in 2020. He will bring us the talk, Interventional Psychiatry, Why Now? So without further ado, let's welcome Dr. Nolan Williams for his wonderful talk. Nolan.
Thank you. Yeah, I changed the title and kind of
you can see.
Neurostimulation approach we've been working on at Stanford. These are my disclosures. To give a little background on neurostimulation generally, and appreciate Mike and Dan going before this, and I'm sure they gave a lot of that same background, and then specifically on theta burst, and then what we've been working on. So as I'm sure has already been mentioned before, I've been very focused, and a lot of the folks on this the symposium have been very focused on treating treatment-resistant depression. The reasons are obvious if you look at the numbers. Quite a few folks in the U.S. and worldwide experience depression. It's the leading cause of disability. It's incredibly expensive, no matter what country you live in, and the treatments that we have available are quite slow and not completely effective for everyone. There's a lot of trial and error sorts of clinical work where folks are having to try a bunch of different oral antidepressants, and eventually they fail enough to end up going to see somebody for an interventional approach. And that's usually around a third to half of people are treatment resistant and don't respond to the conventional oral antidepressants, and those same oral antidepressants in trials haven't really been demonstrated to affect suicidal ideation. And so I've been interested in this problem because we approach depression and more generally psychiatric conditions quite differently than how we approach the rest of medicine as far as increasing the acuity of care. And so, whether it be in China or the US or whatever, as you increase the acuity of a medical condition like a heart attack, right, you have more tests and more treatments. So if I'm having a myocardial infarction and I happen to be in my family medicine doctor's office, we'll have a certain number of tests and treatments. But if you bring me to the emergency room, I'm going to have more tests and more treatments. And you bring me to the ICU, and I have more tests and more treatments. And in psychiatry, as we all know, as you increase the level of acuity, you don't have really tests, biological tests. And on average, you lose treatment options. And a lot of people will say, oh, well, what about electroconvulsive therapy? And that's true in a one-off level if you happen to be the lucky patient who who goes into a hospital and ECT is available in that hospital. But in the US, it's only about 10% of US psychiatric hospitals. I'd be interested, actually, and I don't have the statistics for China, but I can imagine that that not every single hospital in China, or really any country around the world, is going to have ECT literally everywhere. And so particularly in the US, as you increase acuity, you have a 9/10 chance of going to a hospital that doesn't have ECT, right? And so you end up in this situation where you have less options than you had as an outpatient. And that's really a clinical problem that needs a solution. And part of the problem there is that we haven't really focused, like we have in other fields of medicine, in developing acute treatments for these very high acuity, high severity illness states. So somebody with major depression, we know how to treat them as a non-emergency outpatient. When you start getting into the emergency settings, it gets a lot harder, because there haven't been any tools developed really for that besides ECT, which has this access problem. And so, as I said, meds are not completely effective. In the US, about 500,000 people a year are admitted for suicidal depression, and about the average length of stay is about 7 days. And there's a statistic which I think is worldwide, which is that the suicide attempt and completion rate is the highest right after an inpatient hospitalization lifetime. And so those days right after somebody's discharged from their first mood disorder admission is the highest risk of their entire life for completing a suicide. And so when we looked at this data, we said, okay, well, if that's the case, then what is the solution? Well, the solution has to be an engineered treatment that produces a symptomatic elimination, or as many people as possible symptomatic elimination, the suicide risk elimination in a period that is shorter than the average length of stay. So in that case, seven days. So it needed to be shorter than 7 days. And so we said 5 days. You need a day for the person to be seen, and a day for the person to get ready to leave. You're really looking at a 5-day treatment, and it needs to be effective within those 5 days, and it needs to be durable past the 7-day mark. And so if you look at that, things like IV ketamine fall somewhat into that. IV ketamine can reduce people's depression and suicidal ideation symptoms in that kind of time frame, but it's not durable. TMS or ECT can affect those sorts of symptoms. They're more durable, but it's not that quick. And so the current treatment options really didn't fit the bill to kind of check all of those boxes. And the reason why we were so focused on this is because we were looking at this, this particular clinical scenario where people are sitting in hospital emergency room hallways waiting on a psychiatric bed, sometimes a week, without any real access to any treatment that's going to alleviate their suffering, and they're exposed, and everybody sees them there, and they're having the worst day or worst week of their life, and so how can we solve this problem? And that's really what we became focused on. And so we asked the question of, can we engineer a solution, not just to have a serendipitous finding, but can we engineer a solution? And RTMS is an approach that was an engineered solution for a problem thought about in the mid-90s around trying to develop a neuromodulation approach that didn't require a seizure, that didn't require ECT. And that was really one of the goals, I think, for the group of individuals at NIH interested in engineering this solution. And so there was an observation at the time that there was kind of hypofrontality, hypometabolism of prefrontal cortex in depression, on PET scans, on SPECT scans. And so there's this view that that there was an underactivity in that prefrontal cortex, and then there was an observation that in early parametric studies that if you did repetitive transcranial magnetic stimulation, which at the time had been a motor probe, if you did it repetitively in certain frequencies, you could move around this motor evoked potential cortical excitability measure. And everybody at the time thought, man, we can move around the relative activity or predisposition to these systems firing. Maybe you could turn up activity in the prefrontal cortex in depressed individuals. And in the early studies, they were able to see some improvement in depression in individuals receiving rTMS within intramural NIH. And then over time, various groups investigated modulation within the parameter space that allowed for an evolution of this original rTMS approach, increasing the number of days, increasing the intensity, increasing the pulses per session, and found that with that kind of evolution of the parameter space, there was an increase in the response rate that ultimately resulted in this protocol approval by the FDA, which, I'll turn your attention to a few things. This kind of frequency is a frequency derived from parametric studies trying to push motor cortex around. They landed on about 3,000 pulses per day, which in a 6-week course ends up being 90,000 pulses, and that'll be important in a second. And in the original targeting methodology with skull-based measurements, and remarkably, with as little as we knew with this parameter set and this kind of original formula, made about a third of people remit. And it's important to note that the device as a method of treatment delivery is not the treatment itself. The protocols of the treatment. I think a lot of people have been, on the podium saying that, myself and Sean Siddiqui, I think Mike and Dan and others. And so this idea that it's the protocol that's really the treatment, not the device itself. And so with that, various groups thought about a lot of ways to make this parameter space different, make it more efficient. And so one of the ways to do that is to think not just about, kind of deriving parameters based off of parametric experiments, but actually to play brain rhythms back into the brain, right? And so this idea that you could record in a mouse hippocampus and understand the way that the firing rates go there, and then you could play those same rhythms back in to the brain using a TMS device. And so original studies for this in 2005, and what they observed is a dramatic increase in the efficiency of the stimulation. I'll talk about that later, but reducing the time needed and the number of pulses to produce the same changes in cortical excitability. And the way that worked was that they utilized patterns within patterns. And so in the case of continuous data burst, which is inhibitory, it's a pattern within a pattern. So 50 hertz bursts at a interburst interval 5 hertz, right? So you get this high frequency, lower frequency. And the idea is it's like a kind of a AM FM stereo sort of combination. So you have local activity at the really high frequency, and then longer range activity at the lower frequencies. And so that's really the kind of the base idea of this. And with intermittent data bursts, you have an train interval that through various motor cortex experiments was demonstrated to excite the brain or potentiate the brain. And so, it's just another way of looking at that same information I just said. This red circle is, circling the readout of increase in excitability with intermittent theta bursts. And so, humbled to be on this talk with Dan and Mike, and I'll talk about some of Mike's work in a bit. But Dan and Jonathan Downer, and I'm sure he shared this with you, completed a big 400 patient study comparing conventional RTMS parameters with ITBS and demonstrated ITBS to be non-inferior. So a group out of Taiwan did a randomized control trial with 1800 pulses per session of ITBS and demonstrated a separation from sham. And so, ITBS over left DLPFC is antidepressant in a couple of different ways that one can look at it. Very striking non-inferiority study there. This is the parameter space that was approved by the FDA. No change in the schedule, but change in the pulse application to theta bursts, as I described earlier. And I've been using this term pulse potency with this idea that you can increase the kind of power pulse for pulse of the stimulation effect. And so instead of 90,000 pulses to get a course of ITBS, you only need 18,000 pulses. And they used an average MNI coordinate derived from some of Mike's work. And so this is just another way of looking at that. So we became interested in this idea of how do we engineer an approach that can deal with that 7-day inpatient problem within, emergency psychiatry? Can we develop an experimental therapeutic that solves that pragmatic clinical problem? So we went through, and the first thing that we did is we kind of broke things down to the units that we thought were still open questions for discussion around, kind of how to re-engineer RTMS within this time domain. And so the first thing that we looked at was the number of pulses per session and the intensity, and found this great paper from Charlotte Denton-Coven demonstrating 1800 pulses at 90% had the highest change in the motor evoked potential. So we had an idea around our pulse number per session at intensity. And then this sham controlled trial demonstrating a left DLPFC sham separation, 1800 pulses. So we knew the number of pulses per session, the percent of the motor threshold. And then the next question was, we by default have largely a 24-hour inter-session interval, except for on the weekends, which is a 72-hour inter-session interval. But that wasn't conceived of as really a part of that time domain. It was more of a pragmatic decision around how to do this sort of stimulation. But if you kind of re-look at that question, you get into this whole body of literature that called space learning theory that suggests that you really have to stimulate at about an hour interval in between. And so they've done this with psychological experiments. They've done this with mouse experiments. And basically you see, no matter how you slice it, that, if you do less than an hour, you end up with this in mass stimulation, and you don't have incremental increases in dendritic spine enlargement in hippocampal slice experiments. But if you do about an hour, then you have incremental increases in dendritic spine enlargement that keeps happening with every stimulation session. The next, so that's kind of reorganization of the stimulation in time. The second kind of principle that we became focused on was dose. This idea that maybe we're underdosing. The original RTMS trials didn't really do a pharmacology style dose response curve. So as we all know, they've dealt with drugs, the pharmacological evaluations of drugs, they usually have a dose escalation, either a plateau, the behavioral effect, or whatever it is, the kind of readout, and or unacceptable side effects, and that's what your dose range is. And so with RTMS, that wasn't originally done, and there were a lot of concerns around things like seizure. And so, there's kind of signals that may be an issue. And this is one of the trials that kind of demonstrated that signal, which is that they took folks that failed in the brain sway pivotal trial, and they just allowed them to keep getting treated. And then you can see when you get out to 10, 12 weeks, you get a plateau of the effect with more and more dose, and kind of suggesting that that more sessions results in more, more individuals with efficacious treatment. Maybe we hadn't really pushed that dose domain. The problem with doing this with conventional RTMS is that talking about committing people to 3 or 4 months of single daily RTMS, it's not really that practical as a study design. And the third idea, this kind of reorganization in space, it sounds like Dan was talking about this idea too. So I think we're kind of all on this same page of thinking about where these things happen relative to those sorts of concepts. So we know that we're stimulating into a cortical entry point, but really trying to stimulate into a greater brain network. So we became interested in which target one would use and how you would use a target if you were trying to do an accelerated re-engineering of our TMS. And so we looked at this way back when we were trying to develop this technique, and left dorsolateral had the most data. There's this kind of oversimplified principle that if you have a normal mood individual, they're going to have a regulation of the DLPFC to the subgenual, and this is an oversimplification for sure, but useful. In depression, there's an overactivity of the subgenual and under regulation of the DLPFC, and the TMS coil re-regulates that DLPFC subgenual interaction. I'm sure Mike demonstrated this, showed this data earlier, but this idea of if you put the coil in the same skull spot and you look at where it is relative to this kind of ideal brain spot, the closer you are, the better people do. They're closer to this DLPFC subgenual connection. In a retrospective look at it, I think this has been replicated like 7 times or something. And so we decided to do this prospectively. So we used hierarchical clustering to cluster similarly behaving voxels within a region of interest at a correlation coefficient of 0.5 for both the DLPFC and the subgenual. And then we had subregions of both those regions of interest. Then we used the decision algorithm to go through and find the clusters that were big and cohesive and clustered together and anti-correlated with the subgenual. And then that was an automated decision algorithm that then picks a spot that the target is in. And then we utilize a stimulation approach based off of those 3 principles that I talked about earlier: reorganization, space and time, and dose. And this is that protocol. This is another way of looking at it. And really the goal, right? We're giving 5 days, just what we need to give for an inpatient setting, as I described earlier. We're really doing a dose response curve. And in my view of this, we're doing it with, a dose response curve with a neurobiologically relevant kind of arrangement of stimulation and time. And we're doing it at a sufficient dose to plateau the curve. And another way of looking at that, and we published this 3 times now. The first 2 papers, the first one was in brain. It was in people that I referred to as surgical level depression. So there were folks who had met Helen Mayberg's criteria to be in a deep brain stimulation trial. And in my view, because we were giving, I think I showed this figure a second ago, but you look at this like we're giving us the equivalent of a 6-week course of ITBS, pulse dose wise every day. We really had to justify that from an ethical kind of clinical standpoint. So we said, okay, we're going to stimulate in people who, are kind of on the docket to get experimental brain surgery. And, and there's a very small but real bleed risk to any deep brain stimulation surgery. And so this idea that we we're taking folks who are kind of in that range. So, if this did for some reason have an increased risk in seizure, which we have never seen, thankfully, but at the time, we're worried a little bit about that. Then we had some justification to do it. And, quite severe, failed ECT, failed conventional RTMS, many have failed ketamine, psychotherapy, mini meds, and over the lifetime, saw this really dramatic improvement and then a really quick relapse. And everybody said, "Oh, that's very clear. That's placebo effect, and that's that." But I looked at this and I said, "Wow, we've got a signal, and this kind of looks a lot like when folks that I had worked on with cortical stimulator implants had their batteries shut off. So this idea that that maybe we were producing a change in the system, but the change just couldn't be sustained because of the level of treatment resistance of these individuals. And so that pushed me to keep going and to extend this into a less treatment resistant group. Still quite treatment resistant, but about half had failed 5 meds or more. Really, this group wasn't ECT failing. Some had failed conventional rTMS, and what we observed is as one would expect between day 0 and day 1, a decent reduction at kind of a 6-week dose worth of stimulation in depressive symptoms. But if you keep going and you get that second 6 weeks and that third and that fourth, and you get out to what's equivalent to 7.5 months worth of ITBS, you start getting a pretty dramatic reduction in overall symptoms. And it holds much better over time than that original brain letter group. And if you look at the TMS naive people, they get there faster and they hold it for a longer period of time. And you treat them, retreat them, you get there. And we looked at the target positions relative to the F3 EEG electrode spot, quite far away, much further away than I would have anticipated. And so then we did this in a randomized control trial setting. So 22 to 80-year-olds, only in major depression in this case, had to be moderate on the Hamilton Modulus and BDI, and had to be at least moderate or highly treatment resistant on the Maudsley refractoriness scale. And so we were excluding people who had a history of rTMS because of the blind, a history of ECT non-response because of durability issues, and a host of other exclusionary measures. And we found that that this stimulation approach, again in a randomized controlled trial, was clinically efficacious. This is the consort diagram. So 2 people dropped out before they started. One was excluded from the analysis because they weren't transparent with us about their symptoms and diagnosis. About 50% of people were disabled, mid-20s, duration of illness lifetime, and they were about 9 years in the current episode with a mid-30s MADRS score on average. And we had pretty dramatic separation of sham and active at all of the time points, and these are the response rates on the bottom. What was very interesting for us was, if you look, and this is a more atypical way of looking at this, but if you look at individual trajectories and you ask the question of what percentage of people cross the remission line at some point in the 4-week follow-up, nearly 80% did in the active group and only about 13% in the sham group. Now, it didn't hold for very long, and this implies you need some sort of maintenance protocol to really be able to handle all of this. But it suggests that you can move mood in this one circuit in the majority of people, and it doesn't seem to really be, for a lot of folks, it doesn't really seem to be some non-specific sham effect, because we didn't see that very often on the sham group. We did this in suicidal inpatients, and I'll only have a couple more minutes, so I'll bust through this pretty quickly. But 2 previous hospitalizations, 1.5 suicide attempts on average, so pretty sick people. This wasn't prospective, it was just retrospective, just to give people a sense of things. So this is ECT, this is accelerated. We found a really dramatically faster progression to remission in the accelerated folks. And this is the suicide scale scores over time. 6 is relevant suicidality, clinically relevant suicidality, and the durability over time. This is the group pre-COVID. We definitely need to take another picture of some new folks, and some of these guys have moved on in our funding, which we're very appreciative of. I'm happy to take any questions.
Thanks, Nolan. Very wonderful talk. actually the audience can ask questions from the live streaming platform. Actually, I have a question that did you anticipate or did you already done the pre and the post scent treatment? What's the brain response?
Yeah, we've got that data. I don't have it in my slide just because briefer talk, and we're like right on the tail end of getting that put together and publishing it. But I think that I think people are gonna be very excited about this. I'm personally very excited about it. So I think it's gonna be pretty cool. So we'll have that hopefully submitted in the next 4 weeks or something like that. So maybe next go round, definitely interested to show that data for sure. So
cool. Look forward to reading your wonderful papers.
Yeah.
So due to the time limit, so let's thank you again for your talk. Then we will move to next, and maybe at the end of symposium we have some time for discussion. So the next speaker will be Dr. Fei Wang. Dr. Fei Wang is a professor and also psychiatric scientist with expertise in neuroimaging, genetics, mental health screening, and intervention research. She holds the position of director at Early Intervention Unit, affiliate Nanjing Brain Hospital, and Brain Function Institute, Nanjing Medical University. She was assistant professor and associate research scientist of Department of Psychiatry, Yale University School of Medicine. She has published over 100 papers. She is distinguished young scholars from National Science Foundation, distinguished medical expert of Jiangsu Province, the Shuangchuan Program of Jiangsu Province, the NASA Young Investigator Award, and the Clinton Young Investigator Award. Her team is mainly engaged in combining the artificial intelligence for early diagnosis and precise neuromodulation of major psychiatric disorders, including depression, bipolar disorders, and schizophrenia. She will bring a talk about a neuroimaging-based precision medicine framework for depression. So, Fei, welcome.
Thank you, Cao Gan, for your invitation, and thank you for your kind introduction. Let me share my screen first.
Okay, we can see.
Everything's okay? Okay, great, yeah. Great. Okay, it's my great pleasure to be here to present my recent work, neuroimaging-based precision
you can click
framework.
Play to maximize the screen. You can click play.
Okay.
Okay, cool.
Okay, it's full clear. Okay, great. Before I go to details about my current work, I still want to talk about my previous experience. With multimodal neuroimaging techniques, today we talk about a lot fMRI, but for my experience, I took several neuroimaging techniques. During my PhD research with my supervisor, Dr. Dai Zhang, in 2001, we start to focus on schizophrenia and found the focus on frontal functional structural connectivity abnormalities in participants with schizophrenia. Then I went to Yale University in 2004. Over there, I was working with Drs. Hilary Blumberg and John Crystal, focusing on bipolar disorder and schizophrenia almost for 10 years. With the same multimodal neuroimaging techniques, we found pretty similar deficits on frontal, functional, and structural connectivity in BD compared to schizophrenia. In 2014, I came back to my hometown, Shenyang, far away from my current work, Nanjing, and obtained the opportunity to perform the direct comparisons among schizophrenia, bipolar disorder, and the major depressive disorder in China Medical University. Our initial aims, from my experience, I studied schizophrenia, bipolar disorder, major depressive disorder. My initial aim would be explore the specific differences among these disorders in order to identify the potential diagnostic imaging markers. However, unfortunately, with like almost, I hope almost 10 years, we already published more than 15 publications about direct comparisons among schizophrenia, bipolar disorder, MDD. Unfortunately, we only found common changes about neuroimaging feature, like such as frontal posterior functional imbalance pattern, like increased functional MRI features, like alpha in frontal and subcortical regions, while decreased measures in posterior regions, including primary visual, motor, auditory cortices. Unfortunately, we want to see the difference, but unfortunately, we couldn't very much clearly identify the differences among these disorders. These findings suggest that previously I focused on just, I asked Dr. Fox the question, how do you think neuroimaging marker as diagnosis criteria, if they can be neuroimaging marker can be used for diagnosis? My previous initial goal is using this technique to make a diagnosis criteria, but unfortunately, I didn't get it in my last few years' work. This finding make me think about like maybe subtypes with neuroimaging features would be important in psychiatric disorders. Now, let me come back to my today's talk. Sorry, I'm cold. I apologize. Come back to my today's talk. As we all know, the diagnosis of psychiatric disorder remains subjective symptoms and observed signs. Therefore, psychiatrists are struggling to deal with these highly complex, heterogeneous presentations with our symptom diagnostic classifications. Symptom-based diagnosis criteria lead to strong heterogeneity in depression, and consequently, it's very difficult for depression to achieve precision medicine like today's we are talking about the main topic. Traditionally, the idea of precision medicine is to uncover these disease subtypes to improve the treatment outcome. As we can see, precision medicine is already becoming a reality in oncology. They do better than us. That's my idea. However, we are still on the way in psychiatry. One approach to eliminate the heterogeneity with depression is to define it to different subtypes. Psychiatrists have made progress by defining clinical specifiers in DSM system. However, the descriptive symptom features are more descriptive than etiological. As psychiatric disorder become increasingly redefined as brain disorders. Maybe someday our psychiatric disorder just redefined to neurology, or I don't know. If we found some marker to diagnose psychiatric disorder. But fortunately, the MRI technique has the potential of reflecting its underlying neural mechanisms. We suggest that combining neuroimaging data with machine learning approach, depression could be separated into different subtypes. By defining objective neuroimaging marker to define the depression into different subtypes, the treatment accuracy might be improved. So our ultimate goal is to build a subtyping and precise treatment framework beyond symptom. This slide showed our initial work in 2020, combining machine learning approach and resting state fMRI. We identified 2 neurobiological subtypes in major psychiatric disorder, including MDD. Just as I said, when I came back China Medical University, we got the opportunity to collect 3 disorders in one center. After separate 2 subtypes, we named them as archetypal subtype and atypical subtype. Compared with healthy, the archetypal subtype shows similar imaging changes as our previous common changes in 3 disorder comparison, such as significant increased IF in frontal and subcortical regions, while another subtype, atypical subtype, showed significant decreased IF in frontal regions and increased IF in posterior regions. Furthermore, we also do some validation test. We further validated the subtype using cortical thickness, white matter integrity, PRS score, and tissue profile for risk gene expression. We also examined effect of medication status on symptom severity, as they may reflect the pharmacological response of each subtype. In this paper, if you are interested, you can go to 2020, published on 2021, Molecular Psychiatry. In this paper, we first define with IF, 2 subtype, and then using different level data to validate maybe this subtype have their own mechanism. Interesting and thankful, Doctors Winter and Han recently raised methodological questions on this work, which deeply help us to think of subtyping and clustering. They questioned the generalizability of our methods and statistical significance, stability, and overfitting of the results. They also proposed a pipeline for clustering validation to assess subtype validity. Most recently, we responded accordingly with clarification about the disease subtyping, clustering, classification, and applicable machine learning techniques. In the response, we state that it's our opinion. We don't really know which one, which is the best answer to this question. We just state clustering is an unsupervised learning approach, lack of class labels, and there is no universal answer to clustering. Clustering could be measured valid as long as it satisfies our need. By delivering cluster analysis, the psychiatrists aim to disease subtyping. Consequently, to achieve valid clustering, we should re-emphasize the core question generally overlooked. What's the ultimate goal of subtyping? As we stated before, the fundamental problem of psychiatry could be lack of biomarker for developing process treatment. As such, by subtyping psychiatric disorder, we seek to identify an objective neuroimaging biomarker to guide targeted intervention. Interestingly, we found the finding from us like the frontal posterior functional imbalance pattern might be potentially be modulated by TMS. I don't want to spend too much time on these slides. Previous 3 professors have already talked about a lot, but here I want to highlight something like really make me think about my career. As Dr. Blumberg just said, he made a comment in AJP, American Journal of Psychiatry. TMS is positioned to be the antidepressant therapy most likely to achieve personalization. This is consistent with our MRI study findings and its potential for clinical application. Using imaging, we can find where it's wrong, and we want to use some physical technique to just localize that region and give some help for the patients. In this slides, maybe I don't spend too much time on these slides, like Dr. Fox, Dr. Williams, and Dr. Blumberg already said subgenual ACC to left DLPFC. All of their trials have already presented they achieved surprisingly highly clinical effects for treatment-resistant depression. All of these results suggest precise targets guided by fMRI has the potential to achieve precision medicine in depression. But I want to highlight something. It's a little bit different from the LPFC to subgenual connectivity. We also noticed recently emerging studies suggested that the areas within prefrontal cortex, such as the MPFC, Cao Gan just mentioned that reason, OFC and the ventral prefrontal cortex also serve as potential targets for treatment of depression. Furthermore, besides prefrontal cortex, the other lobes like occipital lobe for depression, parental lobe for anxiety, and the temporal lobe for schizophrenia. These provided the opportunity for new targets, TMS targets, combined with our previous findings with defined frontal posterior functional imbalance as an objective neuroimaging marker for subtyping. We collectively propose our this current study. We want to identify neuroimaging-based subtype-specific TMS targets beyond DLPFC to achieve precision medicine in depression. In summary, in this work, our aim is to develop a neuroimaging-based precision medicine framework for depression. We first try to define disease with neuroimaging marker in different centers to replicate our previous work in China Medical University. 2020, 2021 in MP, all the subjects from China Medical University. In current work, we got a collaborator. They provide the new subject from Wuhan University. In this center, we developed the neuroimaging-based subtype and the precise treatment strategy for depression. First, we try to identify fMRI based subtype. Second, we develop a novel machine learning based approach to identify the dominant neuroimaging deficits as subtype specific TMS targets. Finally, in my current affiliation, like Nanjing Medical University, we performed our precise TMS treatment in Center 2. In this work, we focus on depression in young. Like this is very difficult for psychiatrists know, like it's very difficult for treat. And here we go to our primary finding. Our major finding may be 3 parts. First, like we identified a similar machine learning technique, different population. We want to replicate our previous work. Here's in Wuhan University. We still use ELF as measurement. The ELF patterns of the two subtype still 2 subtypes, like archetypal and atypical. So this we still find the increased frontal posterior section, decreased ELF. Here's another subtype, exactly same our previous finding. Interestingly, we also performed the further test on the depression subtypes. Clustering stability test suggests that both subtype compared to that total MDD subtype, subtype performance better than whole sample. After separate 2 subtypes, we want to identify which region is most important region for targets. A reasonable assumption is that effective target would be leading the functional brain regions with prominent differences between depression subtypes and healthy. We chose top 10 regions showing the most significant difference from healthy. Then combining our current neurobiological understanding, we finally identify 2 regions. For archetypal, we choose MDPFC. For atypical subtype, we choose occipital lobe as target region. These slides showed our neuroimaging and both imaging and symptoms both can be improved. Just Chao Gan raised the question. This question has asked me about this one. So in this work, we also analyze the pre and the post, compare pre and post treatment and see if this marker can be used for subtype. We also want to see after process targeting if the imaging marker can be normal, come to more like healthy. If then it may say this method can be more useful. So in this one, we totally 72 hospitalized depressed young. We got a process treatment. Excitingly, both neuroimaging and the symptom improvement were observed after 2 weeks of process TMS. For both subtypes, significant changes in frontal posterior function imbalance were observed after 1 or 2 weeks. in archetypal, I don't go detail. It's just the imaging marker getting better and going close to healthy. And the symptoms, we can also see the symptoms improved. Overall, I want to claim my idea, like if some neuroimaging marker can be diagnosis. And also, I want to use it as a treatment, provide some clue for treatment. Also, we want to see this marker can be used to predict treatment. So we summarize our study in 3 sections. First, again, archetypal and typical seems consistent functional pattern in major depression. Frontal posterior functional imbalance was identified as object neuroimaging marker to guide subtyping and process TMS in depression. DMPFC and occipital might be as subtype specific TMS targets for archetypal and atypical depression. Finally, we all see both neuroimaging marker and the symptom improved in these young patients. It's just like we don't do a double-blind randomized control design. We are on the way to perform the new trial study. Thank you, Chaogan. Thank you for this opportunity. I learned a lot and hope provide some information for this field. Thank you.
Thank you very much for your wonderful talk. So the audience can ask questions in the live streaming platform. So actually I have one question that you stimulated the occipital cortex, right?
Yeah.
For the atypical depression patients. But actually, to my knowledge, I didn't read any little, probably I need to read more, but do you aware any prior literature targeting occipital for treating depression?
Before we decided this region as a target, we read some literature. As I remember, at least 3 or 4 publications, they talk about the visual cortex may be important in depression, and they also target that region for depression. Yeah. We do have some reference. Yeah. In this work, we just separate different subtypes. We think some people like it's similar to Dr. Fox's recent work. They different symptoms maybe have different neurocircuitry, but we didn't talk about the news.
Yeah. So your frontal posterior or frontal parietal. I saw some cysts is on parietal. So why would you choose occipital other than parietal? Because parietal is another hotspot for many studies.
Yeah. Parietal, most of the study I know it's like schizophrenia. They use that region for hallucination, illusion. But for our like emotion regulation stuff, we think maybe occipital is better. And in this talk, I don't have too much time about my animal study. In my animal study, like mice, we also find like visual cortex may be most important in depression symptom. So we just as I said, like we make this decision like like neurology and anatomy and some other issues like choose. And you are right. Maybe parental later we will concentrate on.
Okay, cool. Any other questions from the audience? Daniel, Nolan, do you have any questions to Faye? Okay, so actually, when we are talking about neuroimaging biomarkers for psychiatric disorders, I guess it's a little difficult because the label might be not so accurate. So the doctors will give a diagnosis, maybe depression or bipolar disorder or schizophrenia, and we train the deep learning models anyways. But somehow, sometimes the labels are not so accurate because we know some depressed patients, probably they are bipolar disorders. And so I don't know if we train a 90% classifier, can we trust the classifier, or because just pick up some noise?
I think here's just 1 minute here. Like for my opinion, I think if we do classification first, we have to do clustering or subtype. Like if you do whole sample, it's a heterogeneous. We have to subtype different subtype, and the different subtype might have better machine learning output, like performance.
Okay, cool.
So I don't want to do like a whole sample classification. Have to cluster or subtype first, then classification.
Okay, so thank you very much for your wonderful talk.
Thank you for the opportunity.
Okay, you can stop sharing your screen.
Okay, let me check. Do I turn off my screen? Sorry.
We are still viewing your.
I'm still. Okay.
Okay, cool. So thank you very much for your talk, and now it's time to move to the last talk of this symposium. And for the last talk, myself, I would like to take the advantage to report some of our results. And actually, I'm Chao Ganyan. I'm a professor at the Institute of Psychology, Chinese Academy of Sciences. So today my talk will be the direct consortium and the rest of my time did the project towards neuroimaging biomarkers of major depressive disorder. So like Faye's talk already talked about neuroimaging biomarkers, and actually we are also working on that. But can we get one? It's a still work in progress. So when we talk about the major depressive disorder, so there's a lot of talk about how severe it is, and it can have the suicide risk, and the costs are very high. When we diagnose the patients, usually it's still based on probably DSM-5 or ICD-11, so it's based on symptoms, so still calling for objective biomarkers. And actually, scientists are doing that, so some working on the pro-inflammatory cytokine or HPA axis, or look at cortisol, or look at BDNF. So there's no such biomarker yet. And actually, previous my background is a methodology for brain imaging. So we think about the function of MRI can reflect brain function, and the structure of MRI can reflect brain structure. And as Faye already talked about, maybe someday depression can turn to a neurologist disorder, because we find some biomarker or some circuitry problem, and we have clear biomarker and clear treatment. So probably like Alzheimer's disease, previous is a psychiatric disorder, and because the biomarkers found, move to a neurological disorder. So we think, look at the brain, we can search something like that. However, in the previous studies, mostly have small sample size, residual power, and have flexibility in data analysis, in inconsistent findings, and some statistical issues, and no such neuroimaging biomarker yet. And also we have some studies look at the sample size issue. And recently there's a Nature paper talking about when you're doing brain-wide association studies, you need at least thousands of subjects. So this is an issue. So when we want to find out a neuroimaging biomarker, probably in the future, we should have some big data of maybe brain imaging and plus some deep learning. This might be not the best way, but we think probably it's a feasible way. So this is our lab, what are we doing? But before we get the big data of MDD, so we have to understand the neural underpinnings of MDD, have to do some computational sharing platform. And before that, we should validating from a methodology. So deal with the issues like head motion, standardization, multiple comparison correction. So most prior work is focused on this, and our current work mostly is focusing on the building the MDD imaging database and using deep learning. And previous most work is deal with, we have lots of head motion in the FAB data, and that's artifacts, but usually the patients move their heads much more. So we developed some methodological studies to how to deal with the head motion better, to control the head motion noise. And also our data come from the different sites, and we have lots of site artifacts, how to deal with that. We have some methodological studies, and also for the statistical analysis, there's some paper pointing out that usually the usual conventional way to do the multiple comparison correction has lots of artifacts and false positive rates. So we developed some way to control the family-wise error rate and balance with the reproducibility. So we have some methods, and we want to other people use our methodological suggestions. So we build the toolbox. So there are lots of toolbox in the field, but some of them, lots of manual configurations, and for the big data area, we made some new pipelines. So we built a DPARB, we incorporated our pipeline, DPAS, and adapted our methodological studies, and built a standardized pre-processing pipeline, and we build a platform for data sharing. So each site can process the data locally at the hospital. So raw data doesn't need to go outside the hospital. We have the shared data. We can build a big data consortium. So this is our software is widely used across the world. So we invited the depression research scientists to build a consortium that we can use our software using standardized processing pipeline to process data locally at each hospitals and only share the final results. So we built a ResMeta MDD project. We have 1,300 depressed patients and 1,100 normal controls. This is for the phase 1, and we already moved to phase 2 with a bigger sample size. So actually the idea is each contributing hospital, they use our software to process the data locally at the hospital and only share the standardized process results into our central server. And this process the data has no privacy information of the patients. And also we ensure the data security of the each raw data in each hospital. So we can finally share the final data with all the researchers all over the globe. So we use this as our framework to accumulate the big data. So this is our phase 1. We have the ResMeta MDD project. We have 25 sites, and for the sex ratio, we found out that the female are more prevalent in the depressed patients, and the age and the size. And for the first study, we investigated the default mode network functional connectivity. So what we found is depressed patients showed decreased functional connectivity within the default mode network. But actually, we didn't find anything change in the first episode of drug-naive patients, but rather in the recurrent patients, we found a decreased the functional connectivity with the default mode network. Actually, we didn't find the illness duration effect, but found a medication effect that most recurrent patients, they are using medication. And later we all we also used the longitudinal data to find the medication do have impacts on the depressed patients in the default mode network function connectivity. And then we also extend beyond the default mode network connectivity. We also found the visual network like a Fei already demonstrated stimulating occipital can be a target for depressed patients. So the visual network has deficit in the depressed patients, even in the first episode drug naive patients, and also in the somatosensory and the dorsal attention network. So this is our main results from the data. And from there, so we are collaborating with Dr. Liani Schumann. She is a chair of Enigma MDD. So we want to test if there is any ratio or cultural difference for the brain mechanism in depressed patients between the Eastern and Western. And we just don't want to publish only one paper. We just want to promote collaboration. So we have several training conferences, training the analysis from the hospitals, train them how to analyze brain imaging data, and ask them to submit proposals to analyze the shared data. So this is approved proposals, and some of them are asking, for example, we found the function connectivity of a depressed brain will decrease. So then how about the topological properties of the network? So we found decreased global efficiency and local efficiency for the brain network. So this is led by Dr. Yang Hong from the Zhejiang University and publishing last year Molecular Psychiatry. And some other teams from Hunan province, they look at the dynamic function brain networks. And some other look at the biotypes of the default mode network. And then some look at the disruptive hemispheric connectivity, and some look at some subgroups of the brain patients. And we openly shared the phase 1 REST meta MDD data. So it's already openly shared. So if anybody interested in, you can use this openly shared data to look at the mechanism of the depressed patients. And there are almost over 200 applications approved. And basically we have the data shared. And now because we just moved beyond the resting state data, we also have structured data. So we called it Consortium Depression Imaging Research Consortium. So we recently published a review paper on the psychoradiology. So we discussed what we found and what our next step will be. For example, we look at the MDD abnormalities in different ethnic groups like Chinese and Caucasian, and what factors can contribute such differences, and also look at the surface-based analysis, and also across different mental disorders, like Fei also look at bipolar disorders and schizophrenia. So in our next stage, we also gather the data from bipolar disorders and schizophrenia. And then importantly, what's the longitudinal effects of antidepressants medication and then wet matter alterations. And finally, the question we are very interested in is, can we develop brain imaging-based targets for TMS to better treat depression? So for the first question is, we look at the brain change across different ethnic groups. So we have international collaboration. We have the International Big Data Center for Depression Research invited Enigma MDD working group. So we have the data from China, and then we have the data from Enigma MDD across the world, but mostly it's from the Western Caucasian population. And secondly, we want to go to surface analysis because the Causal paper said traditional volume-based approach has a spatial localization that only 35% as good as the best surface-based methods. So this is they show some results why the surface-based methods are better, and they also showed there are some problems because that's why not most people using surface-based analysis. For example, the relative lack of turnkey tools for running surface-based analysis, or the learning curve is very difficult to learn the surface-based analysis. So because of this, we first build in tools because we need to have the clinic doctors. They can sufficiently, very efficiently use surface-based analysis tools. We build the DPAP itself with based on fMRI preserve ends, and it's central and it's very easy to use pipeline. And we also have the viewers for view the results. So we have these tools. We ask the direct consortium to build the data from China using surface-based analysis, and also the Enigma MD have surface-based data from the Western countries. So this is very initial results, preliminary data. So across the direct consortium, the Chinese patients and Enigma. So when we look at cortical thickness, so for the cortical thickness, we have 5,000 MDDs and 6,000 normal controls. We found the cortical thinning in the posterior single cortex, dorsal medial peripheral cortex in important regions of the default motor network. We also found cortical thinning in the SMA and in the motor cortex. So this is consistent with the previous findings. But the important area, for example, the subgenuine ACC, we didn't find a significant change after correction. So one way we think about is there any difference between the Chinese samples and the Enigma samples? So when we look at the Chinese samples from the direct consortium, we didn't find a significant change in the subgenuine ACC. But when we look at the Enigma data, mostly are Caucasians, we do find the subgenuine ACC showing cortical thinning. But does these results have any statistical significance? So we did a meta regression analysis using the Asian population percentage as a regressor. So mostly direct are Chinese, and in Enigma, there are some sites have the Asians. So we use Asian as a meta regressor. We did find the SGACC showed a significant effect with the Asian population percentage. So that means the subgenuine ACC has some difference in the depression mechanism between East Asians and the Westerners. So we may need to be carefully thinking about the subgenuine ACC as a anti-correlation target to looking at TMI's target. So actually, Shelton is doing work looking at different subgenuine ACC definitions on this very large sample of depressed patients to look at what's a change in the subgenuine ACC function connectivity in this large sample of data set. So maybe in the future, we can find out better way to define the personalized SGACC targets, maybe in the Chinese patients. And beyond the voxel-wide surface-based cross-cultural MDD study, we also trying to look at bipolar disorder, schizophrenia. They are ongoing, and trying to use deep learning to build some biological biomarkers there. So what we think about is we using the deep learning method, for example, for the transfer learning. So for the sex, we can build a classifier with 80,000 samples. But for the depressed patients, at the best, we estimate in the future, maybe we can accumulate 8,000 samples, but still ongoing. But 8,000 is not large enough. So we pre-train a sex classifier on large sample, and using fine-tuning in the smaller depressed classifier, can we have a better classification accuracy? So basically, a student Bing Lu in our lab applied almost all the open data size have 50,000 subjects, 80,000 samples. So what we do is when we do cross-validation, we didn't do the random 5-fold cross-validation because testing sample information will be leaked. So we only use a cross-data size validation. So that means a data set or a scanning set will never participated in the training and testing. So that we want to use the classifier directly applied to some future sets. We never see any data from that set, but we can directly apply the classifier. So firstly, being built a sex classifier with an accuracy of 95%. So model can classify the sex almost all the participants from any scanner without 95% accuracy. So this is pretty sure, and we have many different data sets, never seen the data set to test. And we are very strongly believe we do have this kind of accuracy. And then we also build an online website. You can upload your data to test. And also for the model interpretation, we use an occlusion map, just cover somewhere and see if the accuracy will drop. And if it drops, that means the classifier look at this area very importantly. So what we found is the hypothalamus and some cerebellum regions are very important for sex model. But for sex, we do have some other studies to using brain sex as more information. But in this talk, we are more focused on the disease. So the first thing we tried is transfer to Alzheimer's disease. So we use some transfer learning and use some technique in transfer learning, and also use a cross-site validation. So make sure our Alzheimer's disease classifier can be directly applied in some new site. So what we found in IDNI data site, we have accuracy about 91%. And we apply to some independent data sets. For example, the AIBL or MIRAT. So all classifier never see this kind of data size, never seen the scanner site, but directly applying. So we found the accuracy is about 93%. So we are very confident that all Alzheimer's disease classifier can get information from the brain to indicate if this is the Alzheimer's disease or not. But Alzheimer's disease is too late because we should need some early intervention for Alzheimer's disease. So we directly apply this classifier to MCI data. The classifier never seen MCI before, but it can differentiate who will convert to AD later or who didn't convert. So there's 3 times the predict as AD if they convert. And the classifier never seen Alzheimer's severity like MMC, but the output score has high predictivity with this disease severity. And also the hippocampus is very important in the classifier to showing the Alzheimer's disease. And we also tested different alternatives. For example, in this paper, we use the gray matter volume and gray matter density. If you use gray matter density only, or we use just raw T1 weighted. So all of them can have very high accuracy, but the best one is originally we present in the paper. And also we use the sex as pre-training, but can we use age as pre-training? We can use age as pre-training, but the accuracy is best than for the sex. But still, Alzheimer's disease is important, but it's not of that kind of interest like a psychiatric disease. So we want to transfer to depression, but the initial results is disappointing. Like if we use cross-site cross-validation, that all classifier can directly apply to some new sites, it's not far away from the random accuracy. But if you use a 5-fold random cross-validation, that means in the future, the site need to provide some data to fine-tuning. The accuracy is 70%, but it's not still good enough. So that's why we discussed with Fei that is there any true biomarker in the brain, but maybe because depression is the heterogeneity is pretty high, so maybe subtypes probably better is a better way to build a neurobiomarker. And another way is our lab is looking at the psychological perspective in depression. We focus on the rumination side. And Xiao built some rumination paradigm in F-scanner, and the psychological process is a rumination is a negative side about the past, about self-speech kind of thought. And Xiao looked at that what the brain responds to rumination. So he separated the default mode network into the default mode network core part is more related to self, and the medial temporal lobe more about past, and the MPFC one. And he found during rumination, the core part and the MTL subsystem will have enhanced functional connectivity. So this kind of phenomenon we have observed in many different data sets. So that's why in the future we can't focus on the MTL system that because rumination have enhanced connectivity between the self and the memory past in MTL. And also he observes between the core system and the DMPF system, there's a reduced functional connectivity. So we had some very initial hypothesis is because of core system and the DMPFC, the present system has decreased functional connectivity. So the patients cannot focus on the present. They have the depressive rumination. So one way is we can use meditation. Meditation will focus you to focus on present without judgment, and that can reduce this kind of circuit and break this circuit. Another way, probably using TMS is a good way to break this kind of circuit. So that's why we are trying to using this rumination paradigm to identify the circuit and use some TMS robot in the future to precisely target the same site. But we think that TMS is one way. Another way is meditation. And because we are not in hospital, we should first build some experience with the patients. So we have launched a Mind Flower project, and we recruit patients from the community, and we have a baseline MR scan, and we will give them psychotherapy for the phase 1. It's a cone therapy, kind of like meditation or mindfulness. And in the phase 2, we will give TMS, neuromodulation, and do a follow-up MR scanning. So very initial results, we found we have 8 patients for the psychotherapy, and we found all of them have significant change in the Hamilton depressed symptoms, and also in the anxiety HAMA. So they showed a decrease in anxiety, but actually this study is still ongoing. We are still recruiting more patients and have the therapy and have the brain scan and analysis. And in the phase 2, we will have the TMS to look at if we can change the brain connectivity to improve the rumination, improve the depression. Finally, this is some documentary in our lab work. You can scan the QR code. And if you want to use our software, you can have this kind of free online course from the website. So finally, I would like to thank my collaborators, my students, and the funding sources. And thank you very much for your attention. So basically, this is all of my presentation. So if you have any questions, you can put onto the live streaming platform. And also, I saw Daniel, Faye, Nolan still online. So if you don't mind, would you turn on your mic and the camera? We can have some very brief discussion.
Yeah, sorry, I missed that a little bit.
Daniel.
What did you say?
I mean, we can have some discussion. This is the
Happy to chat
the
about
end of symposium. Yeah.
it.
So.
Whoops.
Hi, Nolan. You are pretty late in your time zone.
What's that?
I mean, it's pretty late in your time zone.
It's later for poor Jeff. I think it's probably like midnight in Toronto, right?
Okay.
Yeah, it's almost midnight.
Thank you very much for
Wow.
staying with us.
Thank you.
No, thank you for the great symposiums, wonderful work that you're putting together. I was intrigued by the potential differences in connectivity in between Asians and Westerners. That we just really highlights the need to personalize our treatment delivery.
Yeah. I do think so. So, but the one thing I'm still, lots of people are thinking about this. Usually for patients, we only have 8 minutes resting state scan. And when we do the function connectivity analysis, 8 minutes usually you cannot have very stable or very reliable. So for example, these 8 minutes can have some peak here, and that 8 minutes can have some peak elsewhere. But NOLA has lots of that kind of work. So NOLA maybe can provide some insights on 8 minutes. Is that enough to target the individual spot?
Yeah, we've been in the current studies, we're getting about 24 minutes of resting state
Wow.
data,
Cool.
and we're really trying to answer that question. We're also scanning people every single day that
They scan every day.
Every day they get accelerated, they get a scan. So they get a scan before we start, and they get a scan every single day, and then multiple times out to the month mark. So I think we have like 10 scan time points, and then we have a very dense kind of targeting scan of 24 minutes. And so the goal there is to really try to answer this question that you're putting up, which is how stable this is and how much time you actually need to be able to pick out a pretty stable scan and a pretty stable target. And so I think we're hoping with that level of density of scan acquisitions, we'll be able to answer that and answer the question of, does this target move around in space over that period of time?
So in science, that the patient will be stimulated 10 times per day, right? 10 hours.
Yeah, no, this is a very hard study to do, and I don't think I'll ever do a study like this again, because we're bringing them in and we're scanning them before the 10 hours, right? So they're kind of there for 11 hours, with us. So they're there, pretty early, and they're getting their scan, and then they've got their 10-hour block of stimulation. And so I think they're getting getting to the scanner around 6:30. They get scanned around 7. They get rushed over to get set up, and then their first RTMS sessions around a little before 8, and then they're done at 5. And so it's very involved. I mean, I've been very impressed with the people that have come through so far, just because that's they're very committed to coming in, basically because of the density of the scanning. So it's we're hoping to answer this question.
Cool. So actually, we are thinking about in our lab to have some kind of like a follow your studies, but I think that for inpatients it's better that you have 10 hours, but
You're right.
Our lab that ask them to come for 10 hours, probably the feasibility is nearly really need the collaboration with the patients.
Yeah, I mean, when I first, I told Mark George that we were going to do the 10 hours or whatever, and he told me no patient will ever do that, but the patients actually don't mind. The hard part, and I think Dan and I have talked about this a little bit, the hard part is the employees that are applying the stimulation over this huge blocks of time. It's that's the part that's harder. I have a strategy on that, but it's tricky. It's really hard to have people that work for you that are going to they're going to be there that long. But I think the patients, for them it's 5 days, and they're happy to be there all day long and all that good stuff. But the employees.
Once they're, I think that once they're in for the day, it's pretty easy. Another couple of hours doesn't make a difference. But I agree with Nolan. The challenge that we run into in our all-day accelerateds is we can't get the employees to stay those extra couple hours. We have to limit to 8 hours. We can't do 10 hours like Nolan.
Oh, I also always, sorry interrupt you before everyone leave. I also always ask myself, like, do you think those important or location or precise medicine most important for these people? I noticed most of the publications, they don't do like precise medicine versus traditional TMS. What do you think? I didn't perform this trial.
Yeah, no, it's definitely a tricky question. I mean, we did what we did, and then what ended up happening was we had, we started out trying to build an accelerated protocol, and then right around the time when we were working on that, Holly Listenby and the National Institute of Mental Health put out a list of kind of necessary components to your neurostimulation grant application. Involved having an imaging based target methodology, and I said, well, gosh, I want to get an NIH grant, so I guess we're going to add to the to this protocol to make it kind of in line with what NIMH wanted. So really, NIMH is the reason why our accelerated, this kind of SANE or S&T or accelerated protocol is the way that it is with the neuroimaging, because they put this out, and then I was like, we're gonna have to go back and get this integrated into the whole approach. And so it's good because we have this package of things. It's bad because it's like grandma's spaghetti sauce or whatever the equivalent is in China, but like this idea that you make this recipe and it's good, but you don't know if it's the tomatoes or if it's this or if it's that.
Exactly.
It's driving the effect, and that's been our problem. And there's enough interest, I think, to go back and try to sort that out. But I guess I always looked at it like the original deep brain stimulation experiments for Parkinson's, right? They were very focused on getting it into the exact right spot within the subthalamic nucleus,
right?
Enough, so you're giving it all day, and you're giving a physiologically relevant stimulation approach, one that seems to, in the case of Parkinson's, seem to kind of abort the tremor, reduce the tremor or whatever, and so I came into the TMS space really thinking for a long time about invasive neuromodulation, and kind of took that frame of thinking into TMS when we first started working on these accelerated protocols. I was really thinking about it in that lens, and that was the lens that that we kind of built this off of. And so that then becomes tricky. You can't kind of, it's hard to split them off. What I can tell you is that I'm pretty sure that the dose piece really matters a lot. Like if you look at it, we have people that come through, and they don't really look any different until, like, we've had a couple of people where several years ago when we were running the original open label trials, where I was like, there's no way this person's getting any better. Thursday afternoon, they don't look any different. They look like literally the same as they walked in. And then it was like the middle of Friday afternoon and they remitted. Like, and it was, and if you would have asked me back then, Thursday afternoon, like, bet whatever amount of money that this person is going to be remitted on Friday, I would have told you no way. Like, they literally looked like the same. Their scores hadn't changed one bit. Then and then they and then they remit the next day. And it was weird. It wasn't something I would have expected. So the trajectories and the dose requirements for people is just so different. We had some people, even in the most recent randomized control trial, where we had this, some of these delayed effects in older adults where they wouldn't get better until a week or two after the cessation of stimulation. And so there's also this component of time, for some people, so it's tricky. It's a really tricky thing. And a lot of it, I think, now is trying to unpack those principles and really kind of dissect some of this out and really figure out what part's contributing to what. But I'm pretty convinced that it really is, we've really truly been super underdosing TMS, and that we need to be giving a whole lot more stimulation than we really ever have previously. So
cool. Thank
Okay.
you.
So maybe more than 10 times per day?
Yeah.
well, maybe more than
Let's
maybe
try.
yeah, maybe more than 5. I mean, for some people, more than 5 days. I mean, for the folks that have failed ECT, and we're in the middle of I think this is in review, or we're resubmitting it now. But we had these folks that we treated in that original brain letter that I talked about for 5 days, and they relapsed really quickly. We gave them 5 days, then a break, and then 5 more days. And then it extended the durability. It doesn't do anything about the degree of remission. You can get them there, but they just don't hold it very long. They relapse out really quickly. But if you give them 2 weeks and you extend the time, if you give them 2 weeks and then you give them conventional or TMS every day after these 2 50-hour blocks, it extends the time further. If you give maintenance accelerated, it extends the time further. And then we've been trying to work with invasive companies to build implanted stimulators that can apply this chronically, right? But this idea of being able to give people a lot more dose in that way, it's not, I think 10, Dan was saying, I'm saying, I don't think you give more sessions in a day. I think we're maxed out on the kind of per day stimulation, but at least for a non-invasive, externalized device, but the number of days, the number of overall pulses per week or month or whatever, I think that we've got to figure out ways to expand that, extend that, figure out what that really is, what it requires.
Thank you. Cool.
I think this kind of line of work will bring lots of hope to the patients, especially for those frustrated their medication resistance. I saw some of these kind of patients and their parents, so they are so frustrated.
I think it's a frustrating problem, right? Like I'm sure Dan's seen this too. I mean, like with standard TMS or standard, whatever it is, people get TMS and they get better. And I've had a subpopulation of people that kind of gotten mad, where they're like, why wasn't I offered this like 5 years ago? Like, I'm better and I have no side effects. This is great. But like, why didn't somebody tell me about this 5 years ago? I spent 5 years like running through all these medications, and I could have just done this and gotten back to my life, and it's just a tricky time, I think, because of that.
Okay, so Daniel, do you have more comments?
No, I think the tricky piece with Nolan's intervention is dose is one part of it, and the other part is the targeting. And so is it the dose or is it the targeting, and would we get the same outcomes if we didn't do the individualized targeting? So there's that personalization piece to the treatment that I think I would probably be on the opposite side and say that I think probably the targeting is the special sauce to the treatment rather than dosing.
Oh yeah, and I'm not taking up because I think it's all you know, I guess my statement was I think you have to have the right dose and the right timing within the target. You know, so I do think it probably ends up being the whole thing.
Yeah, hope so.
Do you have any sorry do you have any plan to perform the child study compare persons like targeting or traditional we just use the LPFC or whatever?
I haven't at one point, and I think Mike jumped off of this. Mike was telling me he was thinking about doing it, but I haven't myself planned to do that. I think I'm kind of you know, NIMH is you know, hasn't expressed an interest in that because they have their kind of you know, target engagement model, and so their view is that's kind of your gold standard or whatever. And I'd encourage people to do that, but yeah, I haven't really myself gone down that route.
Oh, you encourage me. I will try.
Yeah, that'd be great. Yeah, if you want to, yeah, I'm happy to talk offline if you want to try to do something like that.
Yeah. Sometimes I'm afraid it's negative result. I don't know. Thank you.
Okay, cool. So I saw one of the live streaming platforms. One of them have almost 3,000 audience. I don't know, is there any questions from the 3,000 of audience? But actually, I don't know how much we are on other live streaming platform. I will think this symposium is a success. So I would like thank you very much for giving your wonderful talks. And I do hope in the future we can have more collaborations. Just not just simply give talks, but in the future we can build some collaboration. And I think from the Easterners and the Westerners, we have Chinese samples, we have Chinese TMIs targeting, and united with the United States and Canada, so we can have a better picture of the depression and better treatment, maybe across cultures or across race. So hopefully in the future we have more collaboration. So I would like to thank you again and thank you the audience again. So this, we conclude the symposium. So thank you very much.
Thank you.
Thank you.
Thanks very much for organizing. Nice to meet all of you. Take care.
Good night.
Good night.
Good night.
Good day.
Bye-bye.